Hi Claire, how are you doing? I see you're reading the draft research protocol I wrote for the first-in-man study. Hope everything's clear. If not, don't hesitate to tell me.
Hey Peter, it's funny, I was just about to call you to discuss the protocol. Do you have a minute?
Sure. How can I help?
Well, as you know, this is completely new for me, so I might ask you some pretty dumb questions.
There are no dumb questions, Claire, only dumb answers.
Okay, well, I wondered, we are going to develop compound ITL-1437 to treat neurological diseases, right?
Absolutely.
I found this paper that describes how most candidate drugs for neurological diseases fail during development. Even big companies struggle with this.
Yes, but so far we've done pretty well at Innovatica Therapeutics. Overall, our success rate from first-in-man to registration is approximately 30%.
But that's for compounds to treat infectious diseases. According to this paper, the average success rate for compounds to treat neurological diseases is much lower, only 7%.
I didn't know that. ITL-1437 is the first compound we're developing for neurological diseases.
The development of one single compound costs almost €1 billion. So if that's the case, we can't afford too many failures, can we?
You're right. We need to find the rainbow trail and follow it to the pot of gold.
But how do we find the right trail? Do we have some kind of GPS for that?
I'm afraid we don't. Nobody does. We all do the best we can. Basically, we all use the same standard route, which takes us from phase 1 to phase 2 to phase 3.
The same process for so many different compounds and so many different diseases and so many different patients?
Yes, but the devil is in the details, of course. Every trial is different. But you're right. In principle, we all use the same standard linear route.
It's not very innovative, is it? For instance, let's look at ITL-1437.
Okay.
For the compound to work, it needs to reach a therapeutic concentration in the brain, right?
Of course.
So if ITL-1437 doesn't pass the blood-brain barrier in patients in sufficient amounts, then it has no future and we'll no longer invest in it.
Yes.
There's still some uncertainty on this issue. Like, the animal data are promising, but they don't provide solid evidence that ITL-1437 will pass the human blood-brain barrier in sufficient amounts.
Clara, I just don't see where you're going.
So why don't we try to find out earlier in the first-in-man study?
If it was that simple, we would certainly do it. Unfortunately, it's far from simple.
There is a way, though, to find the information we need. If we develop ITL-1437 labelled with isotopes, then we could quantify the amount that reaches the brain by using a PET study. And we could easily include that in the first-in-man trial as a sub-study.
Sorry, Claire, there's no way we would do that. Producing labelled ITL-4037 is expensive and would delay the project by at least 6 months. And it will cost us €2 million extra. We'll stick to the plan and assess the efficacy of the new drug later in the phase 2 studies with patients. Remember as well, this is a commercial company. This isn't a university where science leads. At the end of the day, we need to make money. I'll send you my spreadsheet with the project valuation using net present value calculations. I'll also include a valuation of your idea, so you can see how much it'll reduce the value of our development project. Your MPV will be lower. It's always nice helping young academics find their feet in the real world. Calculating costs and revenues isn't something they teach scientists at university. The sooner we introduce them to the tough entrepreneurial world, the better.
Good morning, Judy.
Good morning, Claire.
Peter. Thanks for the spreadsheet.
You're welcome. Did it make any sense?
Yeah, I studied it yesterday evening, and yeah, I agree. The project value drops by 15 million euros if we include a pet subsidy in the first-in-man trial.
Well, Claire, I'm glad we're agreed.
Well, not entirely. You see, your calculations assume that the product will be developed successfully. And if that's the case, yeah, of course, any extra sub-study will only cost money without providing any added value. But the situation is completely different if we find out in the subsequent expensive trials that the drug does not penetrate the brain adequately and we don't see any clinical effects. Then we lose a lot of money.
Well, that's part of the game. Sometimes you win, sometimes you lose.
I understand that, but the point is we would lose much less if we were able to do it earlier in clinical development by including a pet sub-study in the first-in-man trial. Look, I did some calculations. If we follow your clinical development plan and the product fails in phase 3, then we lose 74 million euros. But if we include a pet sub-study in the first-in-man trial and we see the product doesn't reach the brain, we only lose 8 million euros. It's a difference of 66 million euros.
Claire, you seem to make an academic exercise out of everything. The probability of this theoretical situation actually happening is very low. Just do what you're paid for. Show that our new drug works in patients.
Yes, but I think...
Now, please excuse me. I have a deadline to meet.
Now this is interesting. We all know the success rate for developing new interventions is very low, and all we do is calculate the net present value, assuming the product will be developed successfully. There must be better ways to calculate the project value and incorporate the probability of different scenarios. I think I found myself a challenge.